Welcome to biology. We'll be starting by discussing the nature of science, since biology is obviously a field of science. So we'll go through some basics of what science is and how we explore scientific processes and so on. So what is science, first of all? Well, science is simply a logical way of thinking about the world or looking at things that you see every day. So scientists use observations about the world around them to make inferences, which are logical guesses or predictions. So an observation is something that you can detect with your senses. You can see, you can smell, you can touch something, and then an inference is a logical guess or a prediction from that observation. Science is a constantly changing field, and it's always growing as new information becomes known. So you've probably heard of the scientific method, but what exactly is the scientific method? It's simply a term used to describe the way in which scientists gather and use information. So these graphics show you an overview of the scientific method. We start with observation and then questioning and research and making predictions, then an experiment, drawing conclusions from that experiment, replicating the experiment over and over again, and then reporting the results. So in detail here, we're going to state the problem or question based on observation. So an observation is made, and that leads to a problem or a question that the scientist is planning to solve. Step 2 is gathering information or researching what has already been explored on whatever this problem or question is. Step 3, forming a hypothesis. A hypothesis is a guess based on what you already know. Step 4 then is to test the hypothesis by making an experiment. And then from that experiment, we collect and analyze data. From that data, we draw conclusions about what we've learned from the experiment. And if the data does not support the hypothesis, a new hypothesis is made and the process starts again. So if the original guess, the informed hypothesis, was not correct, that's okay. Scientists don't mind not getting things right the first time. They go back and they do a different experiment, come up with a new hypothesis and try that to see if if the data supports their hypothesis. And in order to make sure that those conclusions are correct, they will repeat the same experiment over and over and over again. And other scientists might repeat the same experiment to make sure that those conclusions are valid, either that they don't support the hypothesis or that they do. Both can be replicated. And then step 7 is to share information with others, reporting the findings. And usually that's done in some sort of journal or publication. So you use the scientific method every day. It's not something that's just pie in the sky, very famous professional scientists do. This is something that you use every day. Here's an example. You get home from a friend's house, put your key in the lock, and you can't open the door. So there's something wrong. Here goes the scientific method. This is your observation. Obviously, you are unable to open the door. So that leads us to a problem or a question. Why can't I open the door? Gather information. Your key isn't turning, so gathering information such as this key worked the day before, or I can put the key in the lock but it doesn't turn, or any number of things that you might gather as far as previous information. So you form a hypothesis. Maybe you think that you used the wrong key. That's going to lead you to an experiment where you use a different key. And then you're going to collect data from that. If the next key does turn in the lock, then that's going to be an observation from your experiment, and that is known as data. And that data is going to lead you to a conclusion. Since the other key opens the door, I must have used the wrong key the first time. Therefore, your hypothesis of using the wrong key was supported. Now, you could repeat this over and over again, but you probably won't. This is just something that happens in a split second. You make these kinds of decisions every day, and you follow this entire process without even thinking about it sometimes. So let's get a little bit deeper into the different steps here. First of all, how is a hypothesis formed? As we mentioned, a hypothesis is a testable explanation for a question or problem. It's a logical guess or a testable explanation. So it's something that we can actually form an experiment from. We often use logical reasoning to form a hypothesis. And there's two forms of logical reasoning that we're going to discuss. Inductive reasoning, which looks for patterns or rules in the natural world, and deductive reasoning, which compares new things to the rules of the natural world that already exist. Since this isn't a particularly intuitive thing to understand, we'll go through some examples of inductive reasoning and deductive reasoning so you can see a little bit more clearly what we're talking about here. So in our first example, we might see an animal and say, this animal is a bird and it has feathers. And then we might see another animal and say, that animal I know is a bird and it has feathers. So therefore we come up with a rule or a pattern that all birds have feathers. And so this is developing a rule of nature from many observations. You are seeing a lot of observations and creating a pattern from that. It may or may not be correct. Deductive reasoning, let's say if all birds have feathers, if we've learned somewhere that all birds have feathers, and this thing that I am seeing right now has feathers, then it must be a bird. Likewise, if electricity is needed to turn on the lights and they're not on, then there is no electricity going to the lights. Again, we're drawing conclusions based on reasoning here. They may or may not be correct. If a light needs a filament to turn on and it is not working, then the filament is burned out. So these are examples of reasoning from observations that help us to form a hypothesis. Once we form a hypothesis, we run what's called a controlled experiment. So a controlled experiment is one that only changes one variable at a time. Because changing too many variables in one experiment makes it difficult to understand the results. So for example, if you try to call your dog in from the backyard and you call come and you call treat and the dog comes, then you don't know whether they came because you gave the command for come or because you were offering a treat. There's too many variables in there. So eliminating one of those things will help you to figure out if the experiment is going the way that you think that it should. And there are parts of a controlled experiment. So there are controls, and those are things that you are keeping the same. An independent variable is the one variable, the one thing that you are changing in the experiment. The dependent variable is what you are going to measure as a result. So let's do a couple of examples of these. You are testing the effectiveness of a new medicine to treat strep throat. Some patients are prescribed the new medication and some are prescribed the previously used traditional medication. So this is an experiment. It's a controlled experiment, so we need to figure out what those different pieces of the experiment are that we just talked about. So what is the independent variable here? Well, the independent variable is the type of medication that's being used. That's what you are physically changing in this experiment. What's the dependent variable? The dependent variable is the ability to eliminate strep throat. So how effectively does that medication work? That's what you're going to measure at the end of the experiment. Did it treat the strep throat or not? And what should be controlled in this experiment? Some controls that they run in medical trials include the age of patients. So you don't want to have necessarily a wide range of ages in your two groups. The severity of the strep throat and the number of days that the medication's taken and things like that. So those are all controls that could be used in that experiment. There are lots of other ones as well. Here's another example. The school district wants to improve standardized test scores by teaching algebra in a new way. Some teachers use the new method and some teachers use the old method. What's the independent variable? I'll give you a second to think about it. The method of teaching is going to be the independent variable. That's what's being changed between the two groups. What's the dependent variable? What we're measuring here is going to be standardized test scores. And what should be controlled? Again, there's a variety of things that might be able to be controlled in this experiment. The number of students in each class, the age of the students, how many days of class they actually have before the standardized test, what were their previous test scores. Items like that would be controlled in order to effectively determine whether this new method of teaching was actually making an effect on those test scores. So in summary, the scientific method is a process that starts with observations. A hypothesis is made using inductive or deductive reasoning and is tested in a controlled experiment. Okay, so we've finished our controlled experiment and we've collected some information from it. The information we've collected is called data. Data can be organized into charts, graphs, or tables because when you're looking at large amounts of data, it makes the most sense to look at the information visually in a chart or a graph. Tables can end up being a little bit overwhelming, and so the charts and graphs give a visual perspective on the information that has been collected, and it's easier to see patterns and draw conclusions from charts and graphs. So how is data used? Well, data is divided into two categories. Qualitative data, which is descriptions and words, and quantitative data, which would be numbers or measurements. Quantitative obviously meaning a quantity. For example, if we're looking at this plant here and we're taking qualitative data on it, the leaves are green, the plant looks healthy, soil is damp, etc. Those would be descriptions of the observations that we're collecting. Quantitative data might be that the average leaf diameter is 3 centimeters, the average number of leaves per stem is 2, the height of the plant is 29 centimeters, anything that could create some sort of quantity for us. And then we're going to use that information, the quantitative data, to graph or create a chart. Chart or graph are terms that can be used interchangeably. When you're creating a chart or a graph, the independent variable goes on the x-axis and the dependent variable goes on the y-axis. So if we're looking at our graph here, let's say we were analyzing the effects of salt on plant growth and we were collecting quantitative data by the height of the plant, measuring the height of the plant after we added a specific amount of salt. We're going to have a dependent variable on the y-axis, our independent variable, which is the amount of salt we added. That's what we're physically changing. On the x-axis, we need units for both of those, centimeters here, grams here, and then a descriptive title. So those are all of the pieces of a good graph. And as you can see, this is easier to read than it would be if we just left it in a table. So how do scientists form conclusions? Well, a conclusion either supports or rejects the hypothesis. So we mentioned this earlier. But is our conclusion proof that our hypothesis is true or false? No. Science is not used to prove that something is true. We can disprove something by doing an experiment that shows that the hypothesis is wrong, or we can be fairly certain that our hypothesis is correct by doing it many times repetitively, but science does not ever prove that something is true, because all it takes is one experiment to disprove it. And so it's a very difficult thing to prove that something is true, because you would have to identify that it's true in every circumstance, in every situation, and that there would never be a repeated version of the experiment that would not draw that same conclusion. By repeating the experiment, we can be more confident of our results. And sharing data with others helps the whole scientific community to further our knowledge. So other scientists might repeat the results of our experiment, or they can use that to come up with a new hypothesis and gain more information. So in summary, qualitative or quantitative data can be gathered into charts, tables, or graphs for interpretation. Conclusions can support the hypothesis, especially if the experiment is repeated. When an experiment is complete, the scientist often submits the results for peer review. So this would be the end of the experiment. The scientist has already drawn a conclusion, and they are then submitting those results to peer review. Peers, as you might know, are individuals at the same level of education or specialization. So in your life, your peers are other teens that maybe go to school with you or are in activities with you. In the scientific community, peers would be other scientists that specialize in the same field of science or have the same level of education as the scientist that's submitting the results. These scientific peers review the experiment to determine if there are flaws with the process or the conclusions. So perhaps the scientist did not run a controlled study very well, and there were more variables than that scientists thought there might be. Or perhaps the scientist drew some conclusions that were not necessarily true based on the data, or they stretched the data a little bit to make their hypothesis be supported. And so peer reviewers are always looking for that. If the peer reviewers are satisfied, the experimental results may be published in a scientific journal. And these journals are known as peer-reviewed journals. So as you get further in your educational career, you may be asked to look for articles from specifically peer-reviewed journals. And that's because they are notably more reliable, because they have had many individuals, professional individuals, looking over them, rather than just one scientist submitting results from an experiment. Peer review is one of the most important ways the scientific community guards against faulty science. So this would be one reason why information in a peer-reviewed journal would be a lot more reliable than something that you might find on a blog post, let's say. What is a scientific theory? So we've now drawn our conclusions from the experiment. We've listed them, published them in a peer-reviewed journal. And if possible, as more and more experiments are done on whatever the topic is, a theory could be drawn. Scientists don't use the term theory in the same way that non-scientists do. So you might say, I have a theory that my sister never gets up before 10 a.m. or something like that. We use the term theory very generally in everyday life. Scientists do not. A scientific theory explains a phenomenon and is supported by many different fields of evidence. So it's explaining a situation that you see in the natural world, and it's supported by many different fields of evidence. So lots of experiments and lots of data and conclusions have supported this particular theory. Scientific theories are broad explanations that apply to many situations. Some prominent scientific theories include atomic theory, cell theory, evolution, and plate tectonics. So these would be theories based on lots of data. For example, atomic theory, you can't see atoms, but the data that's been collected by many different scientists has led them to believe that there are atoms in existence, even though we are not able to physically see them. And how do ethics affect scientific processes? So there are lots of experiments that can be done to test different hypotheses, but sometimes ethics prevents some of those experiments from being accomplished. Scientists must consider the ethics of every experiment. And ethics is simply the branch of knowledge that deals with moral principles. So we're now inserting some morality into the scientific process that maybe wasn't there before. Should a particular experiment be done, or is there a moral reason that maybe we shouldn't be testing on the brains of different humans, something like that? There are 3 ethical worldviews that prevail in science. Anthropocentrism. This worldview places the most value on human populations and human welfare. So humans being the top priority in this worldview. If it benefits humans, then it's worth doing. Biocentrism, which places value on each and every organism, including humans. So not putting human benefits above other organisms, but all organisms in general. And then ecocentrism, which places value on the ecosystem or community as a whole rather than individuals. So this worldview would not be trying to do any experiments that upset the ecosystem or change the natural balance. Which worldview does this individual likely have? If they're saying Earth is more valuable than money, likely they're going to be in that ecocentric viewpoint. So in summary, scientific results can be submitted for peer review and may provide support for a larger scientific theory. All experiments must consider the ethical consequences of their processes and conclusions. Okay, so enough about scientific processes. You're here to talk about biology. Well, biology is the study of living things, and it's an incredibly large field of science. In fact, there are many specialized fields within the study of biology. For example, botany, which is the study of plants, microbiology, which studies viruses and bacteria, microorganisms, zoology, which is the study of animals, anatomy, that studies structures of human body and animals, ecology, which studies the environment, genetics, which studies the inheritance patterns of traits, and molecular biology, which focuses on specific molecules and their effects on organisms. But what exactly are organisms or living things? Well, this was up for debate for quite a while. Until 1668, people believed that life arose from non-living things through a process called spontaneous generation. And they believed this because they made observations. They saw maggots on meat, and they thought that the meat gave rise to the maggots. They saw mice eating grain in piles of grain, and they thought that the mice arose from the grain. Francesco Redi, an Italian doctor, proposed a new theory. Redi suggested that the flies on rotting meat came from eggs that were too small to see until they hatched into maggots. Therefore, they did not arise from the non-living meat. He ran a controlled experiment to test his hypothesis. He used several jars of meat, one uncovered, one sealed, and one covered with gauze, just to let air through. So this one was completely open, this one was just covered with gauze, and this one was completely sealed. The flies only appeared on the meat in the uncovered jar. So the one that had air going through it did not have maggots and flies appearing on it. So his conclusion was maggots came from contact with flies. They did not spontaneously arise from the air because the air did not cause the flies. Reddy's experiment led to a new theory. Living things come from other living things. This is known as biogenesis. Other scientists, however, did not believe Reddy and created experiments to try to disprove his results. Louis Pasteur, on the other hand, was a microbiologist that designed an experiment to validate Reddy's results. He was trying to recreate the results and using a different type of experiment. Pasteur sterilized broth in a swan-necked flask, which trapped microorganisms before reaching the broth. And only when the neck was broken off did microorganisms grow in the broth. So we'll watch this little graphic here. This was the swan-necked flask. You can see it's got a specific shape here. He added broth and he sterilized it by heating it. So he killed all the microorganisms in the broth in these two flasks. He removed the swan neck from one of the flasks so that it was exposed to the air, and the other he kept that neck on. And microorganisms grew in that one that was broken off and not in the other flask. And that's because the microorganisms from the air got caught in this tiny section here. So bacteria and other infectious organisms were trapped here. They got caught by gravity and didn't make it all the way down the flask to infect that broth. And so that supported the theory of biogenesis because the microorganisms were creating more microorganisms. The air was able to pass through here. It did not create microorganisms in the broth. So how are scientific theories different from scientific laws? We've addressed scientific theories, but we haven't quite gotten to laws yet. Again, a scientific theory is a hypothesis that has been tested with a significant amount of data. A scientific law, however, describes a pattern in nature. It generally doesn't include an explanation of why. So this is sort of a general explanation of what you might find in nature. It doesn't have a lot of explanation behind it. For example, every reaction has an equal and opposite reaction. There is data behind that has observations that have been made, but this is a general statement about nature. Reddy and Pasteur's experiments provided data to reject the prevailing theory of their day. So the theory of spontaneous generation was disproven by Reddy and Pasteur. So in summary, although many scientists believed in spontaneous generation, two scientists named Reddy and Pasteur disproved this theory. They showed that living things come from other living things, a theory called biogenesis. So now that we know that living things arise from other living things, we need to identify what even makes something considered living. Well, there isn't just one property that makes something considered living. There are actually 8 different characteristics that all living things share. And this isn't a widely agreed upon number. There are different scientists that would say different characteristics here, but these are the 8 we're going to focus on. So in order to be considered living, something must show all of these characteristics. What are these 8 characteristics? Number 1, living things are made of cells. Cells are tiny but highly organized units that can grow, divide, and respond to their environment. So here's a graphic of a plant cell. This is an amoeba. An amoeba is made of one cell, but a human is made of about 100 trillion cells. Characteristic number 2, living things reproduce. They either reproduce through sexual reproduction, which requires 2 parents, or asexual reproduction, which requires only 1 parent. So birds, for example, reproduce sexually, and there's 2 birds involved, 2 parents. Hydra, which is a type of aquatic organism, reproduce asexually through budding. So here you can see that a new individual is forming off of that original parent. One organism producing an offspring is asexual reproduction. Number 3, living things have a universal genetic code. The universal genetic code is in the form of DNA, and DNA carries the blueprint required to give the organism its characteristics. And we will get much more in depth in all of these characteristics throughout the year. DNA is shaped like a twisted ladder, and that shape we call a double helix. This is the code here in these colored portions that carries the information for the organism. Number 4, living things grow. Some just get larger, while others have an elaborate developmental process. For example, frogs have several stages to their life cycle that involves metamorphosis through a tadpole phase and then into the adult frog phase. Sometimes some organisms just get larger, and that's the only kind of growth that they show. 5, all living things require energy. Some get their energy from the sun, and some get their energy from eating other organisms. The trees and grass in the photo here are called producers because they make nutrients from the sun to use for energy. So they are requiring energy, but they're able to make their own from the sunlight. Cows are called consumers because they consume the plants to gain the energy from the producers. They're not able to make their own food and therefore energy, so they have to get their energy from another source. 6, living things respond to their environments. Organisms have to react to changes around them in order to stay alive. These bison migrate to warmer areas when winter snows begin to cover the plants they use as food. So they have a migration pattern here. They are responding to the environment getting colder. 7, living things maintain an internal balance. This balance is known as homeostasis. So just as your thermostat works to keep your home at a constant temperature when the outside environment changes, living organisms maintain homeostasis when their environment changes. For example, when the environment gets too hot, your body produces sweat to cool you down, and it brings your body back into balance. There's all kinds of examples of how your body maintains homeostasis. Temperature is just one example. It also balances hormones and nutrition and oxygen, all kinds of things. Number 8, living things change over time. Groups of organisms adapt through evolution. So whenever we're discussing evolution, keep in mind that we're discussing groups of organisms. One single organism is not able to evolve, but a group of organisms can evolve over time. And evolution in biological terms simply means that a group of organisms changes over time to adapt to their environment. So evolution is simply change over time, and we're going to discuss this more in depth, and I know that evolution sometimes seems like a scary topic, but in reality, scientists use it just to describe how a group of organisms changes over time, over different generations. So you can see here that mutations create variation in these organisms. Some of the mutations are selected against, others survive and pass their traits on. And again, we'll get into more depth in that throughout the rest of the course. So in summary, living things share 8 common characteristics that help them survive and adapt to changes in their environments.